So a reflex or a reflex arc is one in which we don't necessarily need to process information in the brain. Prior to these slides here, we had talked a little bit about the processing of sensory information, and we had talked about how you need the receptor, you need the circuit that leads to the brain, and then you also need the brain itself to kind of put that information into context, right? Our brain has to try to explain the information that it's being given, right? What does this touch on my arm tell me about the space that I'm in? What do I need to do about it, right? With reflex arcs, we are really using the spinal cord as the integrating center. We aren't using the brain, and they're meant to be these rapid, predictable responses to a particular stimuli. So what I wanted to do was just kind of give you guys a couple of examples of these and talk a little bit about how they work. Both of them, both of the examples I'm going to talk about are proprioceptors, or they involve proprioception, which really has to do with the space, kind of the position of muscles and joints and things like that. Okay? And both of these are going to talk about how we use proprioceptors to avoid damage to our skeletal muscles. So I'm just going to give you guys a couple of quick examples here, and then we'll get on with some stuff. We could obviously go into a lot more detail about the anatomy of these, but we're going to try to keep it relatively simple. So the first example that I want to talk about, it is something called a muscle spindle. Let me get my board out. Okay, so a muscle spindle is a type of proprioceptor found in our skeletal muscle that helps to monitor the amount of stretch in a muscle. Okay, we all know that skeletal muscles can be stretched. We do this all the time. Getting ready to exercise or something like that, we can stretch the skeletal muscle out. And if we don't stretch it very far, it goes back to where it's supposed to go to, right? That's one of the characteristics of skeletal muscle. But it's also possible for us to stretch a muscle so far that we damage it. It has to heal, right? It happens a lot. We kind of talk about that colloquially by saying that we pull the muscle, right? And we would definitely want to avoid that. So these muscle spindles help us to avoid damaging stretch to a muscle. So imagine we've got a muscle here that is not being stretched out. We've got some muscle fibers in this. Muscle spindles consist of sensory neurons that are wrapping around fibers in that muscle. Okay? These muscle fibers are not contractile fibers. Most of our muscle fibers are contractile, really short muscle contracts. These particular ones are not contractile. They're called intrafusal fibers. So we've got this sensory neuron, and those dendrites of that neuron are wrapped around muscle fibers deep inside of that muscle. So something like this. If we were to blow up this area and look at it a little bit more clearly, if we were to take a little section and sort of blow it up, what we might find is that we would have these dendrites kind of coming down, and they form this kind of spindle-like structure. And then in the middle of that, we would find a non-contractile muscle. Those are intrafusal fibers. Okay? So they might look something like that. So this muscle at this point is not stretched out, right? It's just kind of sitting there, contracted, but it's definitely not stretched. Okay? So what this muscle spindle is doing, these dendrites are receiving kind of constant stimulus and it's sending regular action potentials to the central nervous system. Okay? So we see that this neuron is sending regular action potentials to the synapse. So, we were measuring this over the course of time. Okay? We were measuring action potentials over time. We might see that we have an action potential here, we might have an action potential here, and so on and so forth. Okay? So even if we're not stretching the muscle out, this neuron is kind of constantly stimulated and it's sending really regular action potentials to the central nervous system, right? Just so on and so forth. Have a little dot to say that that's what it is. Okay, but now we're stretching the muscle out. Okay, something is now pulling on that muscle, causing it to stretch out. Imagine that you've got a rope or something like that, right? When you pull really hard on a rope, what you see is that all the fibers that make that rope up, they pull together very tightly, right? And they squeeze together, right? Muscles do the exact same thing. And if I suddenly were to pull on this muscle and stretch it out, what I'd see is that all of those fibers would stretch, but what they would do is they pinch these muscle spindles, right? So now all of a sudden, okay? That changes the frequency of action potentials to the nervous system. Okay? What does that do? Okay. Well, there's a couple of things that happen. And this diagram kind of shows it. It's showing the same thing on both sides, but it's showing you how we tested on this side, and I'll explain that in just a minute. So here's our muscle spindle, and it's in our quadriceps. And we notice that there is a sensory neuron, as my diagram shows, it goes to the central nervous system. That sensory neuron splits, and it has axons that talk to motor neurons in a couple of different places. The first set of motor neurons that it goes to, you'll notice if you follow this one out, that motor neuron goes to the quadriceps. The other motor neuron, follow it out, goes all the way out here to the hamstrings. Now the hamstrings are the antagonistic muscle group to your quadriceps. Right? So if I'm sitting here like this, and I contract my quads, I do this, right? Okay, extends my movement. But if I contract my hamstrings, I do this, right? Okay, so they're antagonistic to each other. So in this example, my quadriceps are what are being stretched, right? So something is pulling those out. So if I don't want to stretch something too far, what do I need to do? I need to contract it, right? I need to shorten that muscle so I don't stretch it out too far. So that motor neuron that goes to my quadriceps is going to tell my quadriceps to do just that, contract so that they shorten, okay? But I don't want anything to impede that contraction. I want it to go as smoothly as possible. So what's simultaneously happening is I'm shutting my hamstrings off. Okay? My hamstrings get shut off. So I'm stimulating these guys, shutting these guys off so that they can't do anything. All right? So that's why I have two motor neurons. I have one going to the quadriceps that tells them to contract. The one that goes to the hamstrings is not stimulatory. It actually tells them to essentially shut off. So we call that reciprocal inhibition, and what that does is it just makes sure that I can contract this with the follow. You probably have tested your muscle spindle activity at some point in your life because you've had your reflexes tested at the doctor. And one of the most common ways that we do this is by hitting your patellar tendon with that little hammer that the doctor has, right? You notice when we do that, my patellar tendon's right below my patella. When I do that, you get that little kick, right? You don't have to hit it very hard to get the kick, right? So what's going on, right? My patellar tendon is essentially my quadriceps tendon, okay? It comes down here over my quads, and then it wraps around my patella. It attaches right here at the head of my tibia, okay? So when I hit it, with a hammer, what I'm doing is I'm causing it to pull on my quadriceps, right? It's nothing that can actually damage it. The doctor, as you are well aware, doesn't have to hit your patellar tendon very hard. But what it's doing is it's kind of causing a really sudden stretch in your quads. Okay? That stimulates the muscle spindle to change its frequency of action potentials, sends that message to my spinal cord, and my spinal cord says, well, let's contract the quads. And what do? I kick, right? I kick. Every time, right? Continuously and predictably I kick. But I'm also, and you don't realize this, but I'm also simultaneously turning off my hamstrings so that they can't move. Right? So what that does is hopefully I pull my muscle back, shorten it before that stretch becomes damaging. Okay? Again, this isn't something that could actually damage it, but there are things that theoretically could. Does that make sense? Okay. So, like I said, there's a lot more names we could give to the different fibers and stuff like that in the muscle. I'm not too concerned about that. I really want us to kind of get a broad overview of what's happening with this particular reflex. But it's definitely interesting. Just to kind of pay attention here really quickly to some of the anatomy. See back here. Zoom in a little bit. So again, both of these are showing the exact same thing. You'll notice that this pathway right here, that axon of the sensory neuron is synapsing directly with the motor neuron. This one is a monosynaptic sort of a situation here. Whereas on the inhibitory side that goes to our hamstring, we do have an intraneuron in there. So what that does is we have a synapse right here, and we have a synapse right here. That one is a polysynaptic sort of a situation. So a couple of different examples right there that we see just in this single example. It's kind of a neat situation that we find ourselves. So this is an example of a reflex that is so predictable and so easy to access that it's typically what we use when we test for your reflexes, is we test the ability of your muscle spindle to avoid this damaging stretch in your muscle. Again, you don't need the brain for this. Reflexes are typically really quick, especially these types of reflexes, these spinal reflexes, because we don't have to send information to the brain, right? So, even though action potentials travel really, really quickly, the time it takes to send the information to the brain and then back down to the muscle through the spinal cord adds time to whatever it is that we're doing, and that definitely could have an adverse effect on us. These are really the only monosynaptic reflexes that we have, by the way. The rest are all polysynaptic. So this is about the only monosynaptic reflex that we have. One more example before we quit of reflexes. This one is also kind of an interesting one. We have these things called tendon organs that essentially do the exact opposite of spindle organs. Tendon organs are working to avoid a damaging contraction. Okay? You might say a damaging contraction. It is theoretically possible that you could contract a muscle so hard that you could pull the tendon from the bone. It happens, and it's not a good situation. It almost always requires surgery when it does happen. So what we want to do is we want to avoid that from occurring. So tendon organs are named because their dendrites are actually found in our tendons. You'll notice right here at that patellar tendon beginning is where we find the dendrites of that tendon organ. But what happens is when the muscle is contracting and it contracts too strongly, action potential sent to the spinal cord will turn off the quadriceps, and they'll actually turn on the hamstrings to help to stretch that muscle out and hopefully help to avoid a contraction that's so damaging. All right, so you can kind of see it's the exact opposite. Because we're turning the hamstrings on, so to speak, in this situation and causing them to contract, this is called reciprocal activation. So kind of an interesting thing, right? So our skeletal muscles in our bodies are innervated by motor neurons that arise in our spinal cord. And you can see here how they might be controlled. Several fields will use these muscle stimulators. Some of you have probably had muscle stimulators used on you for different reasons. But usually what they are is they're an electrode placed directly on the muscle, right? And so what you're doing is you're using an electrode that you place directly on the muscle to get it to contract. So in a way, that electrode is taking the place of a motor neuron, right? You're stimulating the muscle directly rather than using a neuron to do that. When we use those muscle stimulators, what we're doing is we're essentially bypassing the Golgi tendon organ. Right? The Golgi tendon organ only works if the neurons that innervate the muscle are involved in the process. But if you use a muscle stimulator, you're kind of bypassing the Golgi tendon organ. And most of the muscle stimulators we use today are relative. Now, one of the things that we've been talking about over the last couple of days is this whole idea of sensory perception, right? And how we have things like our special senses, our eyes, and our ears, taste buds, and our sense of smell. We also have the sense of touch. We have pain receptors, and all of this is not to mention all of the internal things that we're detecting as well that we're not really aware of. They just kind of happen, in our subconscious. So, if you add all of those sensory receptors up, there's a lot of sensory receptors in our body, some of which we're aware of, some of which we're not. But if we add all of those up and we look at where they're kind of located, we find that 70% of our sensory receptors are found in our eyes, which is massive, right? That's really disproportionate. But our eyes are so critical to everything that we do, right? Our eyes are critical for sensing light to help to regulate our circadian rhythms. And then, of course, there's the whole idea of vision as well. And if you think about how much of a role vision plays in your life, it's really pretty remarkable. So it makes sense that we would have a huge amount of our sensory perception devoted to eyesight. Okay. In addition to that, if we take a look at our brain, we see that roughly half of our cerebral cortex is devoted to some aspect of visual processing. Your entire occipital lobes are related to visual processing, as well as part of your parietal lobes, and so on and so forth. So your brain is also devoting a huge amount of its energy into visual processing as well, and forming a picture of the world around you, which is really pretty remarkable. So with that said, let's go ahead and kind of take a look at what we have. So we're going to kind of divide this into, from an anatomical point of view, we're going to divide this into two things. We're going to divide it into the eyeball itself, and then we're also going to look at the accessory structures that help our eye to do its job. I'm actually going to start with the accessory structures, and then we'll take a look at the eyeball itself after that. We'll get through part of this today, and then what we'll do is we'll finish it next week. But let's go ahead and take a look at what we have here. So, as far as the accessory structures of the eye go, these are them in a nutshell. We have your eyebrows, your eyelids, and then we have a structure called the conjunctiva, and the lacrimal apparatus, and then the extrinsic eye muscles as well. So let's go ahead and kind of start to march through these just a little bit and take a look at what we have, and then we'll go from there. So the eyebrows and the eyelids, right? You may not necessarily think that your eyebrows have a huge role, and I suppose maybe for us they don't so much anymore regarding the function of our eyes. But one of the things that our eyebrows do is they work to help to shade our eyes just a little bit. If you go outside on a really bright day, you might kind of do this a little bit. What you're probably doing subconsciously is you're creating a little bit of shade over your eyes to help to protect from that bright sunlight. Obviously, we've created things like sunglasses and stuff like that kind of do a lot of those jobs for us. But if you think about this from sort of an evolutionary point of view, we go back to our ancestors, pre-sunglasses, this was probably a little bit more of an important function for us to be able to function. Another thing that our eyebrows do is they work to catch sweat from our forehead. When the sweat hits that, what it does is it tends to route some of that sweat around our eyes rather than having all of it go into our eyes. Again, if we think about this from kind of an evolutionary point of view, what this does is it prevents that sweat from obstructing our vision, right? If our vision is that important to us, not only is it helping us to potentially catch prey, it's also helping us to not be eaten. And the last thing on earth that you want is for a saber-toothed tiger to get you, right? So if you have something in your eye or something obstructing your vision, like sweat or sunlight, that's going to cause problems, right? So we want to prevent that from as much as possible. So the eyebrows play an important role. Our eyelids also are important. Your eyelids, of course, have your eyelashes on them. Your eyelids are technically called palpebrae, which is kind of a funny word, but they're just really thin folds of skin that you can close over your eyes. You obviously know that you do this when you sleep, but you also know that you do it when you blink your eyes every few seconds. The purpose of blinking your eyes, of course, is to spread lubricant over your eyes. If you've ever been in a staring contest, you know that if you don't blink, your eyes dry out really, really quickly, right? And that's kind of an uncomfortable feeling. So we're kind of constantly producing lubricant over our eyes, and as a result of that, we have to spread it around, and that's why we blink our eyes. One of the things that we do as physiologists, kind of somewhat unrelated, but related to what I have on the slide, is we think about water loss and water gain. And I'll talk about this more when I talk about the kidneys towards the end of the semester. But the whole idea with your kidneys is to maintain water balance. And you probably know this, right? When you drink a lot of water, you have a lot of water going in, you have a lot of water going out, right? You pee more when you drink more. But we also lose water from other places as well. One of the places that you actually lose water from is that it evaporates off of your eyes. If you've ever gone outside on a cold morning, you've put on sunglasses, and the sunglasses immediately fog up when you go outside. That's because the water evaporating from your eyes condenses on your glasses, and that's why you can't see. But you lose a surprising amount of water every day evaporating off of your eyes, something like 100 milliliters or something like that, which is a lot, right? If you look at a water bottle, 100 milliliters is like that much water, which is a lot of water evaporating off of your eyes over the course of a day. So again, the next time you're in a staring contest, think about how fast your eyes dry out and how you're continuously lubricating them in order to help this dry out. Because we do have to lubricate them, we have glands called tarsal glands that are part of our We have a larger gland as well. We'll talk about that in just a moment. That also helps to produce lubricant in the eyes as well. Our eyes are very sensitive to touch. Not only do we have pain receptors on the eye itself, and I'll talk about those in just a minute, you also have sensory receptors in your eyelashes as well. So when something touches our eye, we become acutely aware of that really, really quickly. So again, it's an effort to protect our eyes from damage. So your eyelids, or palpebrae, kind of right here, your eyelashes that are associated with those. The conjunctiva is kind of a transparent mucous membrane that's continuous with the inside part of your eyelashes. So you'll notice that your eyelash kind of folds over, or your eyelid kind of folds over right here, and then it kind of comes down and it covers the eye like this, and it's just sort of this continuous mucous membrane that covers the eye. It's transparent. You can't see it. In fact, if you look at somebody's eye, it's not something that you can see. You see right through it. As a result of that, these models don't really show a conjunctiva on them because it is so thin and transparent. It would be really hard to depict on these models. But it's just a protective membrane that lays over the surface of our eye. You'll notice here that it is divided into a couple of different regions. The palpebral conjunctiva, that relates to the fact that it is kind of continuous with the eyelid or palpebra up here. You have the bulbar conjunctiva and then the conjunctival sac down below. We really aren't aware that this thing even exists until it becomes irritated, right? And then it becomes really, for lack of a better term, an irritation. What's it called? Do you know what it's called when the conjunctiva becomes irritated and inflamed?
Conjunctivitis.
Conjunctivitis, yeah. It's amazing how that works. It's amazing how that works. Conjunctivitis. Yeah, you can just check that. Had me in 50A. Apparently, I say anatomists are not entirely unreasonable people a lot, so they're keeping a list of how many times I say it this semester. I'm already up to a few, and I said it way more than what you think last semester. I know I did. No, really? Was that only eight times last semester?
Well, 4 times.
When I was in college, I had an ecology professor that used dead rabbits. And so we kept a list of how many times in the semester we talked about dead rabbits. I don't know why he did that, but he kept a list of that. So it is what it is. But conjunctivitis is an inflammation of the conjunctiva. This is sort of an umbrella term. Obviously, a lot of things can cause conjunctivitis, whether it's allergies, whether it's some sort of irritation like you scratch your eye or something like that. There's all sorts of things that can cause it. But unless it's irritated, we never are really aware of the conjunctiva. It just is there and it helps to protect our eye. Okay. So the other thing that we see here that provides the majority of the lubricant to our eye is something called the lacrimal apparatus. We use that term apparatus to hint at the fact that there are multiple parts to it. So there's multiple things that make up the lacrimal apparatus. So the lacrimal apparatus is made up of the lacrimal gland and then all of the ducts that drain all of that fluid. Okay, so the lacrimal gland is a really large gland that we see right here, sort of on the superior and lateral side of the eye. And there's multiple ducts that secrete a lubricating fluid onto the eye itself. on all of these models, you can see this really large lacrimal gland on the superior and lateral side of the eye. Those ducts are really, really tiny, but they're constantly secreting fluid onto the surface of the eye. The thing is that that fluid has to go somewhere, right? So what we have is we have essentially a drain in place that works to drain the fluid. And that drain is over here, kind of on the inside part of your eye, or what we would call the corner of your eye, right here, where you have this, and this is going to be more apparent. I know it projects really small. But you have several things right here, the lacrimal punctum and the canaliculus, but ultimately we have them draining into this thing called the nasolacrimal duct, where that fluid is drained into the nasal cavity. Okay? So, as you guys are probably aware, there's you have to blow your nose. Some of the fluid that comes out is because you're draining this fluid from your eye. When those ducts become clogged, the fluid can build up and cause watery eyes and things like that as well. So sometimes we're sick that occurs. Or when the nasal cavity is blocked, this duct can back up and we can run into watery eyes and things like that. So we have a source of lubricating fluid, and then we also have a place to drain that lubricating fluid. The eye muscles, we've already talked about those a little bit, but those eye muscles are going to work to move the eye around in the orbit. There are 6 of them, and 4 of them are what we call rectus muscles. You'll remember from 50A that rectus means erect, right? So the rectus abdominal muscle, or rectus abdominis muscle, is the erect muscle in the abdominal cavity. The fibers run straight up and down. These superior rectus muscle here, inferior, and the lateral medial rectus muscles are right here. And they have muscle fibers that run straight back, right? So when the superior rectus muscle shortens, for instance, what it does is it pulls the eye upward, straight up, okay? The inferior rectus muscle contracts, it pulls the eye straight down. The lateral rectus muscle contracts, it pulls the eye this way. And the same is true for the medial rectus, pulling the eye that direction. Which is all fine and good. We obviously have to look around. We have to move our eyes to be able to see things. But we also don't just move our eyes straight up and down or straight left to right. We also have to be able to rotate our eyes to look over there, for instance, or to look down there, for instance, right? So we have to be able to move our eyes in more than just those 2 directions. So we have these oblique muscles that allow us to do that. Okay? So in concert with the rectus muscle, these oblique muscles can help to rotate the eye. You'll notice the inferior oblique, for instance, has a tendon that originates right here, but the oblique muscle runs kind of under the eye in this direction. So when it contracts, what it does is it rotates the eye, I guess it would be counterclockwise from what you guys would do. And if we're also at the same time using those rectus muscles, we can really move the eye at multiple angles to be able to see things. We need to have an oblique muscle that runs in the opposite direction. The superior oblique originates right here, and it runs kind of like this. But the problem is that's going the same direction. So what it does is it actually turns this way and goes here. What happens is the tendon actually runs through a little loop of tendon called the trochlea, and it runs in that direction to kind of change the direction that muscle pulls. It's kind of an ingenious little thing that we see right there. So the superior rectus muscle, or the superior oblique muscle, runs through that trochlea that you see right there, which is just a little loop of of tendon. It's kind of a neat little thing. And these models, I'll show it pretty well. I'll show it to you in just a couple of minutes. To that end, though, what cranial nerve do you think controls the superior oblique muscle that runs through the trochlea? Trochlear nerve, yeah. Cranial nerve number 4, right? So pretty neat how that works out. Things get their names for a reason. So the trochlear nerve controls the superior oblique muscle, and that also reminds us that it does run through that structure called the trochlea. This view shows that trochlea a little bit more clearly. This is kind of a superior view of the eye. There's an oblique muscle right here, running, and then that tendon is pulled through that trochlea, and that affects the way that it hooks on the eye. So it's kind of a neat thing. Okay. Let me grab. What are we doing? Model. For the eye that's on a green base. Ah, right there. So we have a lot of different eye models around. This is the only one like this that I have on the green base. But we have several that are very similar on the white bases. I think every group has one of those. And then you guys have those individual eye models that are on the wooden bases. One of the things I'll always recommend is, definitely take a look at all the models, right? You may only have one on your desk, but after you feel like you've mastered it, definitely grab another one from another bench, just to make sure that you're getting a good look at it. And you try to see, what can I identify on here versus what can I identify in the other one, and so on and so forth. Part of that is because, we use different models on the tests, and you should be familiar with them all. But it's also kind of reflective of us, right? Like, they all look a little bit different. They're all painted by different companies and produced by different companies. And that makes them ever so slightly different from each other. Even though we're all pretty much the same inside, like our biochemistry is all pretty much the same and stuff like that, there are slight anatomical differences between us inside, just like we see outside as well, right? And so it's helpful to look at more than one example, because if you only ever memorize one thing, and then you're clueless when you look at everything else, then that's not very helpful, right? So we want to make sure that we master this stuff on as many models as possible. So, some of these we have one for every group. Some of these models we don't have very many of them. So we'll definitely have to share a little bit. I'm trying to get our department to save their money so that we can get more of these ones. I really like these guys that are on the green bases. They're just really expensive. This is like a $1,500 model or something. It's really ridiculously expensive. Part of that comes from the fact that they're all hand painted, and what that does is it adds to the accuracy, but also they just aren't very many made, so they're hard to get when they are made. So, I don't know how long this guy was on a waiting list. I think it was backordered for like 6 or 7 months before we got it. So, I'm trying to get us to save our money so that we can have more. But if you take a look, hold it together, you'll notice that you have quite a bit of detail here. There's no eyebrow, obviously, but I think we all know what eyebrows look like. But you definitely can see the eyelids here. Okay, so we have eyelids that are in place. We haven't talked about the eye itself yet, but we'll be able to take a look at it. But we can definitely see like our lacrimal gland right here over on the side, which is going to produce lubricant over the surface of the eye. And then we can see the rest of that lacrimal apparatus here draining down into the nasal cavity right here. So a lot of that is going to be really pretty obvious. We also can see here that we have our eye muscles that are pretty clear. Just a little bit. We have our eye muscles as well. So lateral rectus. We have a superior rectus right here. I'll remove some of these muscles in just a minute so you can see more things. One quick note, really quickly before we get too far into this, you'll notice here on the superior side, there's the muscle, this is the superior rectus right here. There's an additional muscle that most of these models have right above it. So it looks like there's two right here. This is one that you don't actually need to know the name of. It goes to the eyelids, and it's not one that will make you guys know. But it is like right there, and a lot of people sometimes confuse them or try to bundle them into one. But it is two muscles. It's the superior rectus and then one you don't need to know. Most of these models come apart to some degree or another, so you can take this off right here. When we take, if you're not a klutz, if you're not a klutz like me, you can remove this, and then you can look at like the medial rectus and here. Here's your superior oblique. Let me move this in here. So you can see that tendon of the superior oblique right here, running through that trochlea right there, and then continuing on. So you can see that actually pretty well. And so on and so forth. So these models show a lot of stuff. Like I said, there are some things that we don't really have models for, like the eyebrows. But like I said, we all know what they look like. And then, we have kind of the same thing that's in your lab. Who controls what? So this will help us with part of lab 2, but it's also helpful for us to sort of pay attention to this. And then we get to our eyeball as well. So the eyeball, like I said, is kind of a twofold thing. We have the gross anatomy like we're looking at right here, but then we also have the microscopic anatomy that we'll get to on Wednesday of next week. So the eye itself, the eyeball itself, consists of 3 primary layers. The outer layer, the part that we see when we look at somebody else's eyes, is the fibrous layer. It's made of thick, fibrous connective tissue. We'll talk about it more in just a minute. The middle layer is highly vascularized, tons of blood vessels in the middle layer of the eye. And then the inner layer is made up primarily of neural tissue. And this is where visual information is received. It's where vision begins, essentially. Even though the eye is made up of 3 layers, it is largely hollow, and it's filled with fluids that we call humors. We'll get to those in just a little while. And it also is divided into 2 cavities. You'll notice that you have stuff kind of right here, but that divides it into an anterior cavity and then a posterior cavity as well. And you can see that the posterior cavity of the eye is significantly larger than that anterior cavity, which is actually quite a narrow space. So as far as the layers go, that fibrous layer is the outermost layer. Like I said, it's made of a pretty dense connective tissue. It's really quite tough. When you look at somebody, when you look at the whites of their eyes, you're looking at their sclera. Okay? Obviously it's not transparent. It appears white, so it's opaque. It does provide some shape for the eyeball as well as protection. Like I said, it's fairly tough. It's tough enough to anchor all of those exterior muscles that we just talked about. So it's got some strength to it. We used to do cow eye dissections in this class, where we give you a cow eye and have you dissect it. And when you cut the eye in half to be able to see inside, you have to cut through the sclera. And you see just how tough it is. Like you feel that the scissors have some resistance when you're making that cut. It's really, really tough tissue. Because it is just dense connective tissue, you'll notice here on the rear of the eye, this is the optic nerve that would go to the brain, cranial nerve number 2 right here. You'll notice that that sclera kind of continues after it leaves the eye here and here. This connective tissue coats the optic nerve as it goes back to the brain, and it actually becomes continuous with the dura mater, which we learned about in 50A. That's one of the main connective tissue layers surrounding the brain itself. So the sclera is kind of an extension of the dura mater. Okay. In the front of the, oops, sorry, back up a bit. Oh, all right, okay, we're good. Brain farted for a minute there. That fibrous layer does extend all the way around the eye, but the problem is, in the front of the eye, we need light to get in, so we can't have something that we can't see through. So the sclera kind of ends right about here and here, and then that fibrous covering becomes much more clear. We call that part of the eye the cornea. It's transparent. It's still fairly thick and tough, but it's transparent. And the cornea just allows for light to get through, but it still offers protection to the eye. The cornea has a lot of pain receptors in it. And if you've ever been poked in the eye, you know this. It's not a pleasant experience. I mean, in fact, it kind of hurts. But that's because of all the pain receptors that are in the cornea. Okay. So that's the fibrous region. We have the sclera, we have the cornea. We also have the choroid, which is the middle layer of the eye. And the choroid is heavily vascularized. Lots and lots and lots of blood vessels associated with it. The choroid coats essentially the entire posterior part of the eye. It's very, very darkly pigmented. We'll talk a little bit more about the physics of vision next week and how light works. But suffice it to say, it's darkly pigmented because we don't want light to start to bounce around inside of the eye. That would cause all sorts of problems. So the fact that it is darkly pigmented prevents light from scattering around the inside of the eye, which is the anterior part of the choroid becomes quite thick. It becomes the structure that you see kind of right here. It's called the ciliary body. The ciliary body, just kind of a thickened portion of the choroid. It's named because the little projections that come off of it must have reminded somebody of cilia. Either that or cilia got their name because of the ciliary body. I don't know which came first. But nevertheless, those little projections look like cilia that sort of come off of it. But the ciliary body is this sort of thickened ring of tissue, and it has ligaments attached to it. See these little white lines? It has these ligaments attached to it that attach to the lens. Okay. We'll talk about the lens a little bit more next week, but what does a lens do? It doesn't necessarily let light in, but if you're using a camera, for instance, it's like away from our eye for a minute. What do you have to do to the lens before you can take a picture? You have to focus it, right? Yeah, I guess our phones kind of focus themselves these days, but if you've ever used a camera where you actually have to focus the lens, that's exactly what it does, is it focuses light, right? When you look at things, right, if you're looking at something close up, your focus is going to be a little bit different than if you're looking at something far away, right? So what happens is that there are muscles in the ciliary body that are going to pull on the lens, and they're physically going to change the shape of the lens depending on where you need to focus. Okay? Your lens is a solid structure, but it definitely has some flexibility to it. And so when these ciliary muscles that are in the ciliary body pull on the lens with those ligaments, what they do is they change the shape of it, and it allows us to focus on things that are either close to us or farther away, or of course somewhere in between. Right? So it's an important sort of area for us to pay attention to. As we age, okay, our lens becomes firmer and firmer as we age. I have a colleague who likes to joke, the lens is the only part of our body that becomes firm as we age. And I think that's true. Everything else gets softer, right? But your lens of your eye gets more and more firm as you age. But what that does is it makes it harder for you to adjust your vision between near things and far things. So as we age, we often develop nearsightedness or farsightedness, and we need glasses to help us correct it, right? So your vision is one of these things that sort of degrades as you age. But that's because your lens starts to get firmer and firmer. And you can adjust the shape as well. Finally, the most anterior part of this middle layer of the eye is called the iris. And you can actually see your iris, and you look into somebody else's eye. This is the colored part of the eye. This is the round colored part of the eye. And when you look at that, you notice that the iris has a hole in the middle of it called the pupil. Okay? That pupil is what allows light into the eye. So light kind of travels like this. It goes through the cornea, through the anterior part of the eye, through that hole, through that pupil. The lens focuses it, and then we have the light here. And we'll have to see that a little bit later on. Okay. So the pupil is important for allowing light into the eye. And like I said, the proteins that we have in the iris, or that are expressed in the iris, can have different colors. And because they're proteins, that's genetically determined. So that's why eye color has such a strong genetic component, right? Whether you have brown eyes or blue eyes or green eyes or what have you. That's why it has a very strong genetic component. Those are proteins. So if the lens is responsible for focusing light, the pupil is going to determine how much light can get into the eye. Okay? So when we go outside in the really bright light, there's a lot of light available, we can constrict those pupils to allow a little bit less into the eyes, right? But when we come inside to a dark room, we have to do the opposite. We have to allow those pupils to dilate so that we can allow more light into the eye. Right? So when the light's off, see that our pupils are going to dilate to allow us to allow more light into our eyes. So the pupil and the lens kind of work together to help us to form an image or to allow light. This is an example of what pupil dilation might look like. You have a set of muscles that helps to constrict your pupils. You also have muscles that help to dilate your pupils. We'll come back to this a little bit when we talk about the autonomic nervous system, because I want to talk about the parasympathetic and the sympathetic branches kind of doing their job. But we'll come back to that a little bit later. Okay. Finally, we have the inner layer of the eye, the neural layer. It's called the retina of the eye. I'm not going to go into the retina in a whole lot of detail today. I want to kind of save it for next week, but I just want to name it for right now, just so you kind of know what it is that we're sort of talking about. Okay. So with that said, let's take another look at this model, and then I'll let you guys kind of look around using your lab as a guide, and we'll kind of go from there. So let me come over here really quickly. So. Just a hair. Again, most of these models come apart so that we can see around. This one eye kind of comes apart. But let's just take a look before we do that. So if we take a look at the eye right here, you'll notice that you can see the sclera really well. That's the white of the eye. This particular model doesn't have a cornea on it. Some of them do. It'll be just a clear piece of plastic that kind of is over this, but this one doesn't have it. We can, in here, we can see the iris, and we can also see the pupil of the eye.